Welcome to the podcast for Healing Neurology, where we talk about everything that can help heal your neurology, which is really everything from food, lifestyle, and medicine to nature, culture, and politics. There's no topic too big or too small. I'm Jillian Ehrlich, family nurse practitioner, certified in Ayurveda and functional medicine. And we are thrilled about our guest today. Dr. Richard Boles is a medical geneticist and a pediatrician who specializes in mitochondrial medicine, functional medicine, including cyclic vomiting syndrome and other atypical forms of migraine and chronic fatigue syndrome, and autism spectrum disorders. His expertise stems from decades of both clinical work and research at a major academic center, as well as from his most recent experience in cutting-edge biotechnology and genomics. He uses an innovative and integrative approach in both diagnosis and treatment to best serve his patients. He treats children and young adults up to age 25 with a variety of conditions, but his main focuses are on mitochondrial disease and dysfunction, autism spectrum disorders, complicated migraine, including cyclic vomiting syndrome, other pain disorders, and chronic fatigue syndrome. And he is going to blow your mind today. So thank you, Rick. Thank you so much, Dr. Boles, for being with us today. We're really happy to have you.
Oh, well, thank you. And I certainly hope to blow all your minds, but we'll see.
What you know is incredible. And I feel like I end up saying that about a lot of our guests. And I just think we're really blessed to have such incredible minds talking with us. And it's rare to find minds who are willing to go from soup to nuts. You know, usually somebody has their one specialty that they do. They talk about one thing, but I feel like here on our podcast, we can really talk about stress and trauma from the life perspective, from the genetic perspective, just the full range. And so we're so happy to have you to do that with us today about genetics.
Well, thank you.
Let's get started and just kind of start right at the beginning. I'm going to resist singing the Do Re Mi song, but let's start at the very beginning here. Can we just talk a little bit about what is DNA, what is RNA, how do these work, genotype versus phenotype, et cetera?
Okay, well, DNA is in every cell of the body, and it is the entire blueprint of life as to how to make a baby, how to grow a baby eventually into an adult, and everything along the way. So it is all of the instructions that we are conceived with, and there is an identical copy in every single cell.
Great. And what's RNA?
Well, the DNA encodes for RNA. I mean, it's more complex than that, but what you learn in school is that a gene is a DNA sequence, and that gene is transcribed into RNA. So that code is copied into, you think about a similar language called RNA. RNA is another molecule. RNA then takes that code and takes it to the ribosome where it makes a protein. So that RNA code is then translated into a protein. So now you have a protein, and instead of having the nucleotide code of the DNA, the three bases, A, C, T, and G, those are 4 different chemicals, you now have the 20 plus amino acids of protein. So the code of DNA is transcribed into the code of RNA, and the code of RNA is translated into the amino acids of protein. And proteins are what do most things. They're the enzymes, they're most of our structure, they're really what do most of the things in the cell.
And the DNA is contained in the nucleus of the cell, and the RNA is essentially how we get those instructions for the time and place where that cell is out of the nucleus. Is that right?
Well, the vast majority of the DNA is in the nucleus, and there's that one little exception that we will get to, but the mitochondrial DNA, but the 99.99% of it is in the nucleus, and then it is the RNA leaves the nucleus to go to the ribosome, which is in the cytoplasm of the cell, and then there it makmakes a protein. That's and then great. It's much more complicated because the protein has to go through modifications and everything before it finally gets to its form that it's going to be used at and taken where it's supposed to go.
And so can you talk a little bit about genotype versus phenotype? Because we talk a lot about that when we talk about genetics. That's a big part of the conversation.
Well, that's what in school is that you learn is like with Mendel's pea plants is that the gene will be for tall or for short. And then the gene is what the DNA is encoding. And then the phenotype is what the plant shows, if it's tall or if it's short. So you can have a gene that is for tall plants, but the plant may not actually be tall because there's other genes or maybe malnutrition or something else that went on. So the genotype is what's in the DNA, and the phenotype is what you actually get in the living organism, a plant, an animal, a human.
Okay, so our phenotype is like our reality, and it reflects the influence of our genotype, but then it's in light of everything else that happens.
Right. Well, think of it as instructions to build some complex piece of furniture or something. The genotype is the way it's supposed to be based upon the instructions, and the phenotype is what you're actually end out with.
So I'm thinking of myself in shop class, and I'm glad so there's other builders besides me. Okay, got it. And then can we talk a little bit about autosomal dominant versus recessive?
Yeah, there's 2 copies of every gene. Well, let me just say this. There's 2 copies of most every gene. There's a lot of exceptions in genetics. 1 copy comes from the mother and 1 copy comes from the father. So we have 2 complete copies of everything. If a mutation is a variant in a gene that is associated with disease, we try not to use the M word anymore because nobody wants to be a mutant. So sort of not, PC to say people are mutants anymore. So we say they're variants. So variants could be normal, or they can be associated with disease, or they could be risk factor. But in any rate, we're talking about a genetic variant as we're talking about if it's a genetic variant causes disease, people used to use the word mutation. So if a variant will lead to disease, if there is only on 1 copy. So it could be on the mother's copy, it could be on the father's copy, but a mutation or a variant on one of the copy and not on the other. If that leads to disease, it's dominant.
Okay.
In other words, that variant is dominant over the other. You have 1 normal copy and 1 abnormal copy. The abnormal copy is dominant, so it's dominant disease. Recessive is where you have 1 copy and it's recessive. It doesn't cause the disease. 1 normal is good enough. You have to have 2 abnormal, 1 from the mother and 1 from the father, both of them being abnormal. Not necessarily the same variant. It could be another variant in the same gene. That's called a compound heterozygote.
Everybody seems to know about the MTHFR gene. It's a great example because the 2 most common that seem to be checked are the A1298C and the C677T. So if you've got 1 variation in each of those 2 genes, then we say a person is compound heterozygous for those MTHFR genes.
Exactly. Right? And while some disease is dominant and some disease is recessive, it's never quite that clear. There are some genes which can cause dominant and recessive. MTHFR is one of those situations in which 1 copy may predispose towards some things and 2 copies can predispose towards more things. And the other thing is that genes don't cause disease. Genes cause risk for disease. There are people with really horrible mutations that have no disease whatsoever. And so you can pass on that gene to a child who then has the disease. Whether you'll get the disease or not depends on the gene, yes, but many other things, all the rest of your genetics, the environment, and it's very complex. So when we're talking about genes and the relationship to disease, we're really talking risk. Yes, some genes, some variants are really bad and maybe 90% have disease, while other variants may have only a 1% risk for disease. If the disease is 1 in a million, a 1% risk is pretty high, but most people still won't get it.
So, and then what you're saying too is that even if you have variations like MTHFR, any of the MTHFRs, that there's things that we can potentially do about this, and there's ways of kind of changing our health trajectory. Is that right?
I mean, what I do is to try to identify what the underlying genetics are. And I mean plural, because sometimes there could be 1 variant in 1 gene, and that basically causes disease. And that does happen, but that's actually less common. Usually there are variants in multiple genes, some of them more important than others, they're not equal. And all of them, plus other things that we don't know about, plus environment, predispose towards disease.
All right. And then the last concept I just wanted to touch on was this variable expressivity. What is that like? When we talk about genes, what does that mean?
Well, we're talking about dominant disease in general. It's very highly variably expressed because remember, you have 1 variant that's normal and you have 1 variant which is abnormal. And in dominant disease, you could have the abnormal variant, the mutation cause disease all by itself, but there is a normal there. So some people may be completely unaffected. That's called non-penetrance. That's what I said, is that you can have a bad mutation and be completely normal. That does happen. Some people could have the full disease, full penetrance, and they have all the manifestations of the disease. But what's also very common is for people to be partially affected, 10%, 90%, 50%, and several people at 50% may be affected differently because they have different aspects of the disease. So that's what we call by variable expression, is that even in the same family with the same variant or mutation, you may have different family members, everything from the full disease to different parts of it to nothing. And then, of course, somebody with a different variant in that same disease may even be more variable.
Let's talk a little bit about testing. What are the different types of genetic testing that are out there?
Well, genetic testing started with looking on only a part of a gene. Now, genes encode for proteins, and the coding part of the genes are called the exons, EXONS. That's the part of the gene that encodes for the eventual protein. But there are also introns between the exons. So you think about an old-fashioned movie on film, is that you have a few scenes here, then you have a bunch of garbage, you have a scene here, a bunch of garbage. The vast majority of it ends out on the cutting room floor. That's the introns. The exons is what eventually gets spliced together into a movie. The introns are the stuff that are filmed, but you're not going to use. So there's a lot of introns. In fact, the introns are much larger than the exons. And so in a gene, only a very small part of the gene actually encodes for the protein. And then you have the introns in between. So at the very beginning of genetic testing, we would just test for a known mutation, a mutation which is common that causes the disease. There are common mutations for many disorders like cystic fibrosis and muscular dystrophy, et cetera. Many people have different mutations. There, I used the M word again. Okay. Many people have different disease-related or disease-associated variants. But there is one that's more common than the others, and they would test for that. So they'd only sequence that tiny little area. Then later, they could sequence the entire exon. And then pretty soon, maybe around year 2000, it got to the point that you could sequence all of the exons. And so you could sequence an entire gene. And that was like $3,000 to $5,000 just to sequence that one gene. And there's about 23,000 genes in humans. But that was a huge, there could be 30 exons in a gene, and that was a big step forward. So then you started to have gene panels. A gene panel could be 4 genes, it could be 10 genes, small gene panels that cause like a Rett syndrome. There's maybe 4 to 10 genes that are known to cause Rett syndrome, so you can put them all into a small panel. Then people started making larger panels for more complex conditions like seizure disorders, and then eventually for autism. But autism panels were as many as 1,000 genes on them. So then somewhere around 5 to 10 years ago, you had the development of exome sequencing, EXOME. So that's all the exons of all of the genes in the entire human genome. The genome is all of the DNA. So every gene may have up to 30 or so exons. Some may have 1, some may have 70. But you're sequencing every single exon in all 23,000 genes. And that was really revolutionary, and it was extremely difficult to do and expensive and time-consuming to look at. But around 5 to 10 years ago, that became a reality. Maybe 10 years ago more on the research side, 5 years ago more on the clinical side. And that is what most academic centers are doing now, is because you don't really know which genes are involved in most of the time, particularly for something like functional disease, like migraine or cyclic vomiting or chronic fatigue. There's just so many genes involved, and we don't really know what most of them are. So you do whole exome sequencing. A few years ago, whole genome sequencing became clinically useful, and I've been doing most of the time when I sequence patients, I do whole genome sequencing, whole genome sequencing. By the way, that's WGS, S as in Sam, instead of WES, which is whole exome sequencing. Now, the vast majority of the genome, which is all of the DNA, is not the exome. Because remember, the exome is just the exons, the parts, the little teeny parts of the gene that code for the protein. Most of the DNA is in the introns and in between the genes. In fact, 98% of the DNA is not in the exome. So whole genome sequencing is like 50 times more sequencing. You sequence all of the DNA. That's what we do today. So
like when we talk about protein encoding genes, that's 2% of our DNA, and that 98% is, I'm not going to say fluff, but like, I've heard it called dark matter before, just regulatory regions. Regulatory regions. And they're extremely important to determining what gene is on in which cell at what time under which circumstances. Everything from how to make a placenta to how to fire nerve impulses to how sperm will swim is all in the genetic code. In any given cell, the vast majority of it is off.
The rest of the DNA determines what will be on at what time under what circumstances.
So the way I explain this to patients is, I'll tap their knee and say, there's DNA in a cell right here to make an eyeball, but it's really important that that DNA gets turned off so you don't make an eyeball on your kneecap.
That's right. Yes.
So
you need to have the vast majority of the DNA is turned off in any given cell all the time. And even the DNA that is used for that cell, the vast majority of that is only used for certain situations.
And so all that regulatory, what we used to think was not necessary parts of the DNA is very necessary, is what you're saying, because it does the regulatory portions, the on off. So when you're doing whole genome sequencing, you're not just looking at what the DNA that is there, what protein will be made, what instructions, what the recipe is. Are you making banana muffins or cranberry muffins? You're actually looking at when will you make the muffins? When will you do the baking? When will you take them out of the oven? So all those regulatory, what ingredients will go in? Is that what you're saying?
Exactly. And while we know it's important and there are many variants that are associated with disease, we don't understand exactly how to read that code yet in terms of how it does its regulation. We're still learning that.
Just because I know this is in listeners' minds, what about 23andMe or Ancestry? Where did those DNA tests come in?
Well, let me first go over large versus small variants. Okay. Before whole genome sequencing, you had to think about, is a variant small? Is it like the misspelling of the word, one nucleotide? Or is it something that's maybe 10 or 50 nucleotides long? Think about a monk trying to copy every single letter in every book on every floor of a huge library in a giant university. And the monk has to go through and copy all of the DNA to make a baby, in both the sperm and the egg. So you have to copy every single word. If you misspell the word, that's called a missense mutation. Those are small. If you miss an entire word or you duplicate the word, those are still small. You're going to find that only by proofreading. The larger mutations is that a book was missed, a book wasn't copied, or maybe an entire shelf wasn't copied, or maybe one book or an entire shelf was done twice. Those are deletions and duplications. And those were done by completely different technology. You need the sequence to read the code, but you need to have like an organization, like a card catalog or so, to see if you've done every book once and no more than once. So the sequencing, whole exome sequencing, looks for small mutations. Large mutations were looked at by chromosomal microarray. A long time ago, they did chromosome analysis, and that can find things that are millions of nucleotides, maybe hundreds of thousands of nucleotides in size. Then they have these chips that they call microarrays, and they can go down to 1,000 nucleotides, in some cases even 500, in some places 5,000, depending on the part of the DNA. So you have tiny little mutations are done by sequencing up to about 50 nucleotides, and down to about 500 nucleotides or so are done by a chromosomal microarray. So you see there's that area in between 50 and 500, neither of them would do it. And one doesn't count for the other. You're looking for totally different mutations. Mutations can be large or small or in that little gap area. With whole genome, you can do all of it. You get the small, the medium, and the large. So you don't have to have two things. Beforehand, you could do whole exome sequencing and chromosome microarray. And that would give you the vast majority of things in the exons, inside the genes. With whole genome, you get all of it. You get the small, the large. You don't need to do a chromosomal microarray as well. So with whole genome, you also can get mitochondrial DNA and trinucleotide repeats like Fragile X, and there's about 40 others involved in neurological disease. So it really does the whole genome. It does everything. To speak about 23andMe, think about a Lamborghini, a real Lamborghini, okay? And think about a toy Lamborghini. Toy Lamborghini looks like a Lamborghini, and it's fun, and it's a toy, and I like to play with it, but it won't get you anywhere. 23andMe is a toy. It's fun. But you're looking at thousands of nucleotides. But what we're doing whole genome, we're looking at billions of nucleotides. The difference is between thousands and billions, a million times different.
Can you talk about what a single nucleotide polymorphism is?
That's just where one is changed for another. SNP, if people talk about SNPs, it's pronounced SNPs, but it's S as in Sam, N as in Nancy, P as in Paul. Okay. Single nucleotide polymorphism. That's where you have a missense where one variant is changed for another, but it's relatively common in the human population. Sometimes people use a 1% rule, but it's just arbitrary.
You're saying that all of us could really be in the 1%?
Well, the number of SNPs in the average person is in the tens of thousands within and around the genes itself. When I do a whole genome sequence analysis, on the average, 20 to 40,000 changes are noted. Okay. So I get, so I can't look at 20 to 40,000, so I use the computer to get rid of the vast majority that are incredibly unlikely to cause disease, and then focus on the several hundred to a few thousand that may cause disease. And I look at those. But the SNPs are the ones that are common in the human population. So when you do like 23andMe, they only look at SNPs. They look at the things that are common in the human population. Some of those have risk for various disorders. What I look at is I generally look at the exact opposite. I look at the ones that are very rare that might be related to significant disorders, not the ones that may be risk factors for common conditions, but the ones that are likely to be the cause of or a major contributing cause to the patient's illness.
So then tell us about mitochondria, which is one of your specialties, and mitochondrial DNA. This is different than the DNA that we have in the nucleus of our cells that we get from both of our parents, correct?
Correct. And this is something when I was in school, they had no idea that it existed. So I didn't learn it there. I had to learn it later. The mitochondrial DNA is a tiny piece of DNA. It's only 16,000 nucleotides versus what's in our chromosomes. The total is 3 billion nucleotides. I mean, twice, because one from the mother, one from the father, right? So this little 16,000 nucleotide only comes from the mother. The sperm need a lot of energy, but they do not put their mitochondrial DNA into the egg. It's left out. So the egg's mitochondrial DNA comes only from the mother, and that forms the mitochondrial DNA of the child. So our mitochondrial DNA is identical to that of our mother, and all of our brothers and sisters, as long as they have the same mother, our mother's brothers and sisters, and our mother's mother. If we're a woman, we will pass it on to all of our children. If we're a man, we'll pass it on to none of our children. So that's how the mitochondrial DNA gets passed on. Of course, if there's a new variant, then that it will be different. That new variant will come up or maybe be gone. But for the most part, the code is the same exactly between mother and child. It's in the mitochondria, which is in the cytoplasm, not in the nucleus. There are thousands of copies of mitochondrial DNA in many cells. So unlike in the nucleus, where you either have 2 copies, 1 copy, or 0 copies of a variant, for mitochondrial DNA, you have thousands of copies of the variant. And you could have 23% mutation, which is called heteroplasmy, mutant heteroplasmy, or you can have 78 or 99.5% or 0.31%. You can have any amount from 0 to 100%. And so it's extremely variable. And different cells in different parts of the same individual will have different proportions as well. So it does change over time just by randomly which cells get divided and how much mitochondria DNA they have in there. So the mitochondrial DNA only has 37 genes in it, and it makes 13 proteins, unlike the 23,000 genes in the nucleus. It's very important because the mitochondrial DNA is entirely based on energy metabolism.
And is there exchange between our nuclear DNA and mitochondrial DNA?
They communicate to some degree. They don't exchange each other. I mean, over millions of years they do, because many of the genes from the mitochondrial DNA eventually went to the nucleus. The mitochondrial DNA used to be a bacterium that was captured by a cell. And so it is now like almost a separate symbiote that lives within each of our cells, and it has its own complete genetic machinery. It has a very high mutation rate because it's bacterial genetics. So it goes by an entirely different set of rules, but they have to communicate back and forth, and some diseases are due to the miscommunication between the two of them as well.
What are those diseases?
Well, I mean, the mitochondria is involved in energy metabolism, and mitochondria itself are these little pieces of, inside each cell. They're little cellular organelles, and they make the energy for the cell. They do many other things as well, but the mitochondrial DNA is completely involved in energy metabolism. Since neurological cells, nerve cells in the brain and outside of the brain and the nerves are electrical, and they have very high energy requirements. So mitochondrial abnormalities generally cause neurological disease. They can cause muscle disease too, because muscle has high energy requirements. So they say neuromuscular disease. So disorders of the mitochondria generally cause neuromuscular disease. There's many different types of neuromuscular disease because the brain is involved. It can be depression, panic, anxiety, schizophrenia. It can be in the eyes and the ears. Neurology is important, so it can be blindness or deafness. It can be dysautonomias of the autonomic nervous system, which can include things like fatigue and pain, GI dysmotility, cyclic vomiting syndrome, irritable bowel. It can also include the peripheral nerves, and it can cause a neuropathy of different types. So the nervous system is really, when I'm talking about the nervous system, I'm talking about really much the whole nervous system. It can cause epilepsy, mental retardation, developmental delay, autism, ADHD, really anything that involves any nerve cell. And the disorders of the mitochondrial disorders can be due to a mutation in the mitochondrial DNA. They can be due to a mutation in the nuclear DNA that makes proteins that are then imported into the mitochondria. Mitochondria have about 1,000 proteins in it. Only 37 are made from the mitochondrial DNA. The others are made from the nucleus and imported into mitochondria. And it can do with mutations in both DNA. The mutations in terms of the communication between the nuclear and the mitochondrial DNA, those are nuclear mutations that are involved in proteins that communicate between the two genomes. So they make neuromuscular diseases just like mitochondrial disorders in general.
So let's talk a little bit about the modes of inheritance for DNA sequencing through families. How does that happen?
Well, first of all, we were just talking about mitochondrial DNA. So I'll mention that in maternal inheritance, a mother will pass on the mitochondrial DNA to all of her children. Some of those, if there's a mutation that's homoplasmic, meaning 100% of her mitochondrial DNA have that mutation or that variant, then all of her children will get it. Of course, the dad won't pass it on to any of his children. If that mutation is heteroplasmic, meaning only part of her mitochondrial DNA have it, some of her children will have it, and some of her children may not, and they'll all have different percentages. So one person may have essentially 0%, while another one has 50%. Someone else may have 25, someone else may have 90. So it's extremely variable. But the point is in mitochondrial DNA is it's passed on to a woman to at least a lot of her children, if not all of her children. So that's what you see. And you see the females pass it on to their children and the males not pass it on to theirs. So that's the exception. That's that 0.01% of DNA outside of the nucleus. The rest is the nuclear DNA. We mentioned that disorders can be dominant. A disorder that's dominant is a person will have a variant that's normal and a variant that has the abnormality on it. So 50% of the children will not have the mutation, and all of their children will not. 50% of their children will have the abnormality, the mutation, the variant that's associated with disease. But because of variable expressivity and non-penetrance that we discuss, some of those individuals may not have any disease at all, but may still pass it on to people that have disease. Others may have the full disorder, while others are somewhere in between. Recessive is when two people carry one mutation, but it takes two copies to cause disease. So in this situation, the parents are carriers. So the mother's a carrier who's normal. The father's a carrier who's normal. They each carry one copy of the disease and one copy that's normal. 1 in 4 times, both the sperm and the egg will have the copy that's abnormal. 50% for the sperm, 50% for the egg, because they have 2 each. So 25% of the time, the baby will get 2 copies. The baby will not be able to make whatever it is that recipe is for. That protein will not make it, or will make an abnormal protein, and then that baby will be affected. So you find normal families that suddenly have a child that's affected. But once you have a child that's affected, there's a 25% chance for another child affected, because it's 50% for each sperm and 50% for each egg. So that's recessive disease. There's also X-linked disease. And X-linked disease comes usually from the female, but it's a lot more complicated than that. We have the autosomes, chromosomes 1 through 22. These autosomes are identical in that the mother and the father both have 2 copies each, and there's no difference on gender. But then we have the sex chromosomes, the 23rd pair, X and Y. A female has 2 X's, a male has an X and a Y. So a mother who's a carrier will have a disease-associated variant on one copy and another copy that's normal. So half of her eggs will have the abnormal copy. The man will either pass on a sperm that has an X or a Y in it. If he passes on the sperm with a Y, then it's a boy. And if the mother passes on the variant and the father passes on the Y, then this boy will only have one X chromosome, because that's what boys have, and it will have the variant. So this boy will have the disease. The mother is considered a carrier and is usually normal, and that's what you learned in school, but in reality, she may be mildly affected. And an X-linked dominant disease, which is another complication, the mother can be just as affected as the child, but those are rare. So in classical X-linked disease that's recessive, a normal mother has 50% of her boys will be affected, because it depends on which X. Now, if the father has an X chromosome in the sperm, then the girl will have one copy of the mutation, assuming that's what the girl got, and a normal X from the dad, and will be a carrier just like mom, who could then pass it on to her boys. So that's how X-linked disease works. Okay, now the really complicated one. That's all the easy stuff. You learned all that in school. People knew that 100 years ago. This is what I learned in school. Almost no disease works the way that we discussed. Almost all diseases work in a completely different way that you've never heard about, called multifactorial inheritance or polygenic inheritance. Asthma, diabetes, autism, migraine, chronic fatigue. These are all complex but common conditions. They are multigenetic, polygenic, and they're multifactorial, meaning there's also an environmental component. There's not one mutation on one gene that does it. There are multiple variants on multiple genes which confer the genetic part of the risk, and then there's environmental factors that confer another part of the risk. So whether a child has the disease or not depends upon which variants were inherited from the mother, which variants were inherited from the father, and how they work in a very complex interaction among themselves and with environment. So in multifactorial disease, the recurrence risk is often 1 to 3%. It can be higher, it can be lower. It's extremely complicated. But if you look in families, you often will see 1 or 3% or so. But it can be higher, and it's extremely, we call pleomorphic. Different people will have different manifestations of the disease in terms of severity and specific symptomatology. So most common disorders are multifactorial, that there's a genetic component, multiple genetic components, and they run in families, but they're not the nice, easy rules. And there's not just one thing you're looking for on DNA, but I have to look for multiple factors.
So this is really where the puzzle is figuring out which factors are relevant for which patients at which time, and then what is presenting, what has the most strength.
It is a puzzle, and there's really not a clear answer in most cases. And let's say that in a good case, meaning in a case in which what we're looking for, what we're hoping to find, and which is actually pretty common, is that we'll find factors from the mother and factors from the father that may or may not be related to the child's disease. And by the way, the child can be an adult or whatever, independent of what the child's age is at this point. I'm a father, there'll always be my babies. But anyway, I'm going to say child, I'm also a pediatrician. So what will often happen is I'll find variants in the mother and variants from the father that may or may not be related to disease. And I call these candidate variants. And if a candidate variant cannot be tested and it cannot be treated, then it has very little interest from this day and age. Maybe eventually we'll be able to do something about it. But I'm most interested in the ones that can be diagnosed, and in particular that can be treated. So what will often happen is I will discuss the genetics of the family, and I'll say, okay, there are these 10 candidate variants. And by the way, that's not an unusual number to find. This is the likelihood of each one, in my opinion, being involved, and if it's a minor or a major component in my mind. And these are the type of treatments, if anything, I can think of that might help with that candidate variant, if it's involved. And then we discuss the pros and cons of each. And it sounds like that's a really bizarre thing, because I don't even know if they're involved at all. And if they are involved, I don't know how much. And if they are involved to a large extent, I mean, how do we know how to treat them? Mm-hmm. But many times I can figure out, based upon what the gene does, what might be a treatment. We can discuss the pros and cons. And however bizarre it seems, about 50% of the time, I can come up with a therapy that will actually work in the patient. That meaning that the patient takes it and is at least moderately better.
That is remarkable. That is remarkable.
I never suspected that when I first started that. We find a lot of candidate variants, and many of them I can't do anything about. Some of them I can, and nothing happens when you do the treatment, and other ones I do, and the patient gets better. But I'd say overall, about 50% of the patients that I can find something that improves them based upon the DNA alone.
Based upon the DNA. So when you're doing your whole genome sequencing, you're really looking at the introns and the extrons and the clinical picture that the patient presents with and their mother and father's presentation, and then putting together the pieces. You essentially create your own picture about where you're going to try influencing their genetic expression or their phenotype, their presentation.
Yes, and it's really complicated, and I really need to understand the patient extremely well.
Yeah.
I mean, not to say anything against the laboratories, because I was a laboratory director for 6 years of 2 different major laboratories. I haven't been for the last 4 years now. But as a laboratory director, you get a little bit of clinical information. You really don't know the patient. You don't know the family, and you can't go back and get information. So It's not nearly as good as now, where I spend a lot of time talking to the family, talking to the patient, examining the patient, going through the medical chart. And then I have a feeling of who this is as an individual, as a family member. And then I look at the genetics and I say, okay, these are the variants that I think might be related. And then I go back and discuss the family, and that's where the rubber meets the road. That's where it matters, because I have all these genotypes, I have all these ideas, and we discuss it. Well, no, I don't think so. This doesn't really fit and doesn't have this. Okay, we'll throw that one out. What about this one? Oh, wow, that fits completely. My child does have IgA deficiency, or does have brown macules, or there is a family history of pancreatitis, or there are bent little fingers, or something like that. There is a family history of problems with anesthesia. And then we dive into it a little bit more, and we find more and more things that fit. Even though I spent a lot of time talking to this family and putting together this really complex note, I didn't ask most of the important questions, because you can't ask every possible question. So it's really connecting the patient to the genetics. That's where it really is done.
And it's remarkable, because we're looking again at the genetic code. We're looking at the genetic code for clues, but we really need to still customize it to this day and age.
It's a lot of art to it. Yes, it's customizing, it's understanding the family and getting a good history and looking at the DNA. There's a lot to it.
What we're really going to talk about after we've just had this whole genetics discussion is dive into some of the details about how we make this information, what we know about genetics, practical for daily use, for medicine, for health, for wellness, for life. So if we could start out, let's just understand, what is the concept of a functional disorder or a functional disease? What does that mean?
Well, the term functional disease is not the best term, but I'm constantly challenging everyone to come up with a better term. It's used differently in different subspecialties and by different people. Some people think that functional disease means that it's not real, it's in your head, or it's psychosomatic, but it could be farthest from the truth. I mean, that's not what I mean at all. It means that it's based on the function of the cells, the tissues, and the organs, not their structure. So a structural abnormality would be like a heart attack or a bowel obstruction, something like cancer, something that's physical that you can see on a scan that causes disease. So that's structural. Functional problems would be like the heart is just not pumping well enough, or the bowel is not working well. Those are far more common than structural problems. But modern medicine looks for structural problems. And you look to see, what is it? What's going on? Where's the problem? And so you do your MRI scans, you do your endoscopies. And if you don't find anything, then it said, oh, it's functional. Well, that's true, but that doesn't mean that functional is any less important, and it's certainly more common. So functional symptoms would be what you feel, is what is subjective. Pain, fatigue, nausea, dizziness, those are all functional symptoms. It's subjective. You might look at someone and think they look like they're nauseous or dizzy or in pain, but it's subjective in the person and to some degree in the observer as well. Other subjective functional symptoms would include anxiety, depression, panic. So some of them are in the neuropsychiatric range as well. And there's many others that can come up with tinnitus, ringing in the ears, double vision or inability to focus. Those are often functional issues. So those are functional symptoms. When we talk about functional diagnoses or functional syndromes, it's when you have certain functional symptoms that meet a diagnostic criteria that somebody came up with. And they're somewhat arbitrary. Some of them were common ones are migraine headache, chronic fatigue syndrome, irritable bowel. There's hundreds of them, fibromyalgia, cyclic vomiting syndrome. It can go on and on and on, interstitial cystitis, POTS. There's many of them. And a lot of people, including myself, would put major depression and generalized anxiety disorder into that category.
Like brain fog. There's no ICD-10 for brain fog.
Yeah, it's a symptom. Brain fog is really common in many of these disorders, particularly in chronic fatigue syndrome.
When we're talking about functional disorders, we're really talking about how something functions. So the process from a step A to step B. And it's not something that we can necessarily see in a still snapshot like you would if you took a picture, if you did an endoscopy, you took a picture of a stomach and you can visualize an ulcer in a flat, still moment in time. How the bowel moves is going to be one of these functional disorders.
Right. I mean, in GI and gastroenterology, I've been told by many specialists that 90% of what they see is functional. They look for structural problems because those can be serious, a cancer or an obstruction or something. I mean, they can be life-threatening, and they look for that. If they don't find anything, which they usually don't, they say that it's structural. So if you put a camera down there and you don't see anything, that means that it's functional. Right. If you do an emptying study in which you see how well the stomach empties over 12 hours and it empties slowly, but the camera was normal, then you assume that it's functional. It's not functioning right, but you don't see a structural abnormality. If that makes sense. Tests will show functional abnormalities, but it's not the common tests. Most of the common tests won't find it. And certainly imaging tests, ultrasound is unlikely to find it, or MRI. They might find dilated loops of bowel because it's just not emptying very well. But it's often going to be negative imaging studies.
So with that understanding of functional disorders, let's talk about the genes that underlie functional disorders. What's unique about them?
Well, one of the things is that functional disorders run together, that if you have one, you often will have more. There are some people with just irritable bowel or just migraine. And there's definitely people like that, and a lot of people like that. But those are generally not the ones that I'm talking about. I'm talking about people that have severe functional disorders or multiple functional disorders. And those are actually quite common. If you have migraine, you're more likely to have chronic fatigue. If you have cyclic vomiting, you're more likely to have fibromyalgia. If you have tinnitus, you're more likely to have interstitial cystitis. I mean, there's been many studies that have been shown if you have one, you're more likely to have another. There's a group of people that have a very large number. I've seen people with 10, 20. I think the most I've ever seen is somebody that had 30 of them.
Wow.
And it's really sad. But it's the same condition, just in different parts of the body. And then they understand they don't have 30 different problems. They just have the same abnormality of their nerves, and it's the nerves going everywhere. So they have symptoms that are related to different things. It's different types of doctors take care of it because it's in different parts of the body, but it's all the same disease process. So it's not just as comorbidity on the individual level, but it's comorbidity on the family level. If you have any of those functional disorders, your first degree relatives are far more likely to have it. Well, there's migraine with aura and migraine without aura. They're the most common. Migraine without aura, which is by far the most common type of migraine, your first degree relatives are about twice as likely to have migraine. But if you have migraine with aura, they're 4 times more likely. With chronic fatigue syndrome, it's around 4 or 5 times. So your relatives are 4 or 5 times more likely. But there's also, if you have migraine, your relatives are more likely to have irritable bowel. If you have cyclic vomiting, your relatives are more likely to have fibromyalgia. I mean, it doesn't breed true. Is that the genes which underlie the functional disorders, as in part 1, they predispose towards other disease. They don't cause them. And they predispose towards functional disorders in general, not a specific one. Although there's probably other genes that indicate why one person gets migraine, another one gets irritable bowel, et cetera. So you see families in which the family history looks negative at first because everybody has something different. You say, wait a minute, these are all functional. And you say, that you can clearly see that there's a family trait if you view them as functional and look at the pedigree in that way.
So this seems to suggest that there are some specific genes that might feed a lot of these disorders. And what are those genes?
Indeed, that's correct. A lot of them are channelopathy genes. And that's something that I've noticed over the last few years and that I'm actively working on. But most of it is still unpublished, although some of it is. What a channel is a protein that crosses the membrane and it lets an ion through. So these are ion channels. Ions are salts like sodium, potassium, chloride, magnesium, calcium, etc. So a calcium channel, and many of them are calcium channels, will open up and let calcium through. But the majority of the channel is not the donut through the membrane, the hole that calcium goes through. Most of the channel is the bouncer. It says, okay, I'm a calcium channel. You're not calcium. You're magnesium. You can't come through. It has to recognize calcium. So the channel is off. It won't let calcium through. Then the channel is flipped on. It will let calcium through, but only a certain number. And then it'll flip off again. So it's the regulatory part again. It's not just making the channel, but making sure the channel works exactly as it's supposed to in that cell at that time. So a calcium channel will let calcium through. When calcium goes through, it's positively charged. It changes the membrane potential. That cell's more likely to be excited or to, in terms of nerve cells, they send impulses when they depolarize, right? Where they fire an impulse or what happens is when they depolarize, there's a whole bunch of channels open up, including sodium channels, and it all rushes in. So if a channel lets too much in, it's more likely for that cell to depolarize. I find it's leaky channels, leaky calcium channels in particular. Calcium is the messenger for the cell to do its thing. It depolarizes, a gland makes hormones, a muscle contracts, a nerve sends impulses. So if this calcium channel is leaking, then the cell is more likely to be twitchy and more likely to depolarize. Now that you think, oh, I get it now. No, biology is much worse than that, okay? Because each nerve cell in your brain has about 100,000 other nerve cells that synapse onto it. Some of them are sending positive signals, positive neurotransmitters. Some are sending negative signals. So if a channel won't open properly and it's an inhibitory channel, that neuron's also more likely to fire. So you say, okay, well, it's neuron hyperexcitability. Well, okay, it can be. But if it's an inhibitory neuron, a channelopathy may cause the neuron not to fire when it's supposed to. And it's an inhibitory neuron, so other excitatory neurons are unopposed. So you see the complexity, particularly when each cell has multiple neurons synapsing to it with many, many different types of channels. I mean, there's many different types of calcium channels. I don't know an exact number, but it's probably near 100 or more. There's potassium channels, there's sodium channels, and everything. So when one channel is leaky, or it's not working very well, it disturbs the nerves. But certain nerves are more likely because it depends on what channel it is, what mutations in that, and also all the rest of the genetics, all the other 3 billion nucleotides and the environment and developmental issues. So in other words, if you have a channelopathy, it's variable expressivity. And I mean extreme variable expressivity. It can be inherited from a parent who has no symptoms at all. Perhaps because that parent has another gene which makes the disease not occur. It's a protective gene, but the other parent put a vulnerability gene. So there's protective genes that cause resilience, and there's vulnerability genes which cause increased likelihood. So let's say that the child got the channelopathy variant from the father. The father has no problem at all, but the father also has some other genes that protect him. The mother has genes that make it more likely to have disease, but she doesn't have the channelopathy variant. So she's normal as well. The kid gets both the channelopathy variant and the predisposing variant. So the kid has disease. So when you look in families, you see variable expressivity. It's possible for somebody else in the family to have more severe disease. And I see that every once in a while. But usually the patients are most severe because they got all the way to me, the geneticist. So usually they're the worst off. But sometimes there's somebody else that's more severe. Many times there's other people that are less severe. There may be other people in the family with the same severity, but different. You know, really bad irritable bowel instead of really bad migraine, or mild irritable bowel instead of mild migraine. And some people are completely unaffected. But what makes it difficult is a lot of the oligosymptomatic people, the mildly symptomatic people, are really in the normal range. If you have headaches once in a while, if you have diarrhea once in a while, if you have these sort of things once in a while, you are in the normal range. You are normal. But remember, all of us have hundreds of thousands of sequence variants that makes us different. We all have things that make us more likely or less likely to have issues. So even though that you're normal and not diseased, you still have genetic issues which make you who you are. So I know that's really complicated. But what I'm trying to say is that when you look at the family, sometimes you can say, oh yeah, this whole family has chronic fatigue or a migraine. And sometimes that's the case. But the vast majority of these families are much more complex with asymptomatic, mild, moderately symptomatic, and severely symptomatic relatives with different functional problems.
And when you talk about channelopathies, just to go back to that for a second, when we're talking about channels on membranes, we're talking about membranes all over the body. So it could be in your central nervous system. It can also be in a calf or in your toe or in anywhere. Is that right?
Well, we're usually talking about channels that are on the surface of a nerve cell. But that nerve cell can be a peripheral nerve that is in the leg going to the toe, or it can be in the eye, or it can be in the intestine, or it can be in the brain, anywhere in the brain. So functional symptomatology can cause symptoms anywhere. It can cause tinnitus if it's in the ear. It can cause problems with the eye, like focus issues. It can cause pain or vibration or weakness or any sort of thing like that. It can cause dizziness if it's in the ear, because that's where the dizziness is, the semicircular canals or in the inner ear. If it's in the brain, it can cause movement disorders, ataxia, which is like you walk like a drunk or a baby. It can cause issues with, which we think of as psychiatric, depression, anxiety, panic, if it's in the emotional centers. It can cause ADHD if it's in some of the executive centers. If it's in the long pathways that have to do with communication, it can cause autism. So, it can cause developmental problems or even mental retardation in some people, but that's rare. So it can affect anywhere. So then you have a person who has a lot of symptomatology, and feels a lot of things that nobody can verify, and all the tests come back normal that they sent off. And there's just not something right about that person. There's maybe they have some learning disabilities or some emotional issues, or something is just wrong in the head. So you say, okay, this is not real. It's all psychiatric, or it's Munchausen, in which they made it up, or it's psychosomatic, or it's drug abuse, or it's something like that. But it could be farther from the truth. These are suffering people who could have a channelopathy, among other different genetic causes, which are affecting nerves in the rest of the body, causing symptomatology, that the brain sees real symptomatology because the nerves are sending false signals. And then there's also issues in the brain at the same time.
In preparation for this podcast, you and I were talking, and one of the things that I love that you said was, show me a Munchausen's and I'll show you a channelopathy.
Yes. Most of the ones that I have, I've been able to find channelopathies in.
How do you distinguish a channelopathy from essentially a regular problem? I don't know how else to say it, between
it's regular. It's just that people haven't realized it yet. The medical establishment is going to have to come to terms with this whole thing eventually. But yeah, it's really regular. It's really common. But yeah, you mean the type of things that people understand and accept. Okay.
How do I know if I have a headache, or how do I know if it's a channelopathy?
Well, you don't, and it could very well be. But again, that's in the normal range. If you have headaches occasionally, particularly with stress, that makes you human. Mm-hmm. If you have headaches that are frequent and severe and you can't do your job, that's a disease. You are diseased. Mm-hmm. And in that case, what may be the channelopathy needs to be treated, or it would be good to treat it if you can. Where in the first one, you wouldn't necessarily need anything. Although even in the first situation, a lot of people take like coenzyme Q10 or other parts of the mitochondrial cocktail. Even with very mild disease. That's really common in family members. You have somebody that has really severe functional disease, you find a channelopathy, you treat them and they're better. So then there's a brother, the sister, the aunt, the father, the grandmother has mild disease. And so you say, well, we don't want to try this medication. It has side effects. They don't need that for occasional headaches, but maybe some of these over-the-counter supplements that are safe, we'll give those and then the headaches go away. So that's actually really common. And that's because we were able to make the diagnosis because of a more severely affected individual. But in the population, what if that person wasn't born? You still have a treatable disorder in a family, not just a treatable disorder, you would have a treatable trait in a family that really doesn't have disease. So it's complicated. It challenges people's idea of what's normal. There
Absolutely.
is no normal.
There is no normal.
The people who think they're really normal are the scary ones.
Yeah, I agree.
There is no normal.
Those are the people not to be trusted.
Yes, there's no such thing. It's a spectrum across the board on everything. Normal people have headaches. Well, some people don't, but the vast majority of people do. And how many headaches do you have, and how much of a problem is it? We can say that about any symptom.
And so what can be done about channelopathies?
Well, the channelopathies are some treatments that are the same for various channelopathies. there's some treatments that are different. So first we'll go on some of the ones that are the same. Many of the calcium channelopathies and at least one of the sodium channelopathies is treated with potassium. Because potassium is not always the most benign situation. I mean, look at a murder mystery. What is the culprit? A lot of times it's someone gave too much potassium. It's got to be done under a doctor's order unless you just want to take guacamole and bananas. That has a fair amount of potassium. Unlikely to kill yourself on that unless you have renal failure. But, it's so a lot of times we get potassium, either increase it in the diet or by prescription and monitoring potassium levels. Certain channelopathies do very well with potassium. Other channelopathies don't, so I like to know the diagnosis first. Many of the channelopathies have mitochondrial dysfunction. I suspect that part of the reason is that if your channels are leaking all the time, your pumps have to constantly pump it back. If you have a calcium channelopathy, calcium keeps leaking, you have calcium pumps that pump it back. The channel opens, calcium rushes through, no ATP, no energy is needed, but you need ATP to pump it back to the other side. It's like a rock drops, so then you have to put it back on the shelf again. Then it drops. It drops on itself once you let it drop, but you need energy to put it back on the shelf. That's where you pump the energy into it to put the calcium back on one side, and then it just floats to the other side from a high to a low concentration. So if the calcium's leaking all the time, you need the pumps are working 24/7 to put it back, and that takes energy. There are probably other connections as well. In particular, calcium is a trigger to turn on the mitochondria. The fight or flight response involves calcium to a large degree. You turn on the entire cell, including the mitochondria. What parts? Pretty much all of energy metabolism you need to treat on. If you see the tiger face to face, you need that ATP to survive. That's what it's meant for, the fight or flight response, to survive a situation like that. Or another, caveman is coming up with a big club and he's much bigger than me. You need the fight or flight response. So the calcium channelopathies very frequently involves mitochondrial dysfunction. In fact, when I do that, the test name is called MitoSwab. That's a company name. It's the only company that I know that does this. And then from saliva, they can measure the mitochondria in buccal cells. So you just do a spit. Basically, it's a swab of the cheek, and you send that in, and it tells you how much ATP you're making, how much energy. And in that situation, almost all of the channelopathies, without exception, have mitochondrial dysfunction. So many times I can find mitochondrial genes, many times I can't. But the treatment is to treat the mitochondria. And treatment for mitochondrial dysfunction is to provide all of the micronutrition that mitochondria need. Many of the vitamins, and perhaps most of the vitamins, they do their work inside the mitochondria. That's where they're needed. So what vitamins? A, B, C, D, E, K? I mean, most of the vitamins. What minerals? Magnesium, zinc, selenium, magnesium, molybdenum. I mean, most of the minerals. So they work in the mitochondria. So the first part of the cocktail is to give very high doses of mitochondrial required micronutrition. In particular, in some cases activated, that what's in over-the-counter vitamins may not be the activated form. The second thing is to detoxify the mitochondria, because the mitochondria that aren't working very well, they produce a lot of pollution. It's just like a car that's running, the engine's not running very well. Let's say that it has a high energy demand, like having to pump the calcium. The high energy man would be going fast uphill or pulling a trailer. Or it's just the engine's not working very well. It's not converting fuel that it burns into energy well. So it's either on the supply or the demand side, it's going to produce more pollution. The same thing with the mitochondria. The mitochondria burns fats and sugars. And if the mitochondria are not burning it effectively because the supply or the demand side, you don't burn the fuel as well and you produce pollution. That builds up in the mitochondria and it causes issues, particularly reactive oxygen species, free radicals that some people in this audience may have heard about. So we need antioxidants and other detoxifications to try to detoxify the mitochondria. So I'm talking like carnitine, glycine, and all of the antioxidants. There's many of them: coenzyme Q10, alpha lipoic acid, vitamin E, N-acetylcysteine, riboflavin. There's far more than that. But those are some of the major antioxidants. Oh, vitamin A as well. Beta carotene is a form of vitamin A. So the treatment for mitochondrial dysfunction is to provide the nutrients the mitochondria need and to try to detoxify the mitochondria as much as possible. And so that works in a lot of the functional disorders, including the channelopathies. And some people do very well just on the mitococktail alone. I call it cocktail because cocktail is many things together, not the alcohol. Alcohol is actually a mitotoxin. Some of the audience may have realized that already. And Oh, one of the questions is, why is mitochondrial so important? Why does mitochondrial problems cause so many issues? So we spoke about that, and all cells need energy to do almost everything. So if the mitochondria is not working right, you can get a whole bunch of problems, functional disorders and non-functional disorders.
Let's talk about medications.
Okay. Some of the patients do well on over-the-counter or dietary solutions alone. Eating more potassium or giving potassium by prescription, giving antioxidants or micronutrition, the mitochondrial cocktail. But some patients need more than that. I start with the natural ones first because a lot of times that's enough. And actually, I start with that while I'm getting the DNA sequencing to figure out what the exact gene is. Because the pharmaceuticals or the medications are really dependent on the exact gene. There are certain genes for which acetazolamide, which is also known as Diamox, is used for high altitude situation, and it's a diuretic, are very good for. There are other calcium channelopathies in which propranolol is useful. And then there are channelopathies for other treatments. So some of these treatments, people say, well, wait a minute, a lot of doctors just recommend these treatments, particularly propranolol in general. It's because a lot of it is channelopathy, and a lot of doctors have realized that propranolol often works. There's probably other reasons why it works as well, other than just this one particular calcium channelopathy that I see all the time, which is RYR2, which I did publish that one, ranitidine 2, which is a calcium channel. But there are other drugs. So I'd like to get the DNA sequence. The DNA sequence says, first of all, do you need a drug? Maybe it's potassium you need. Maybe it's riboflavin. Second thing, if you do need a drug, which drug do you need? Do you need amitriptyline, which is another one that's often used? Do you need acetazolamide or propranolol or gabapentin or something else? And then the third thing is, okay, we're going to use this drug, but what dose are we going to use? Are you a high metabolizer or a low metabolizer, fast or slow? If you're a fast metabolizer, a normal dose is not going to really touch you. It's just destroyed by the liver. And so if you had a normal dose before and you failed, maybe you need a higher dose. If you're a slow metabolizer, people say, oh, you can't use that drug. But no, you can use the drug and it's cheaper because you use a very small amount.
Yep.
Use a small amount to get the same thing. So they said, no, I tried that drug before. I tried amitriptyline. I had all these horrible side effects. Well, you have a very poor metabolizer. Did you try like a quarter of the dose or something? And maybe, so that will help you to know which drugs were taken before that really you don't know they didn't work, or a new drug to know which dose to start with and how high to go on it. So the genetics really helps me across the board as to how to prescribe and if I should prescribe.
Can you give us some examples of some specific channelopathies in which you found them in a patient and you were able to treat them?
Yes, I have a patient that I saw many years ago, and I saw him in a cyclic vomiting episode. Maybe abdominal migraines a better word for it because the pain in the abdomen was worse than the nausea or vomiting. But he was screaming and jumping off the table. He was a young adult. And if I didn't know this, and I had seen many other patients with cyclic vomiting, I would have thought the same thing the ERs did, that there's just something wrong with this guy. People don't react like this. He was just screaming and yelling and, really causing trouble. And he was vomiting and had nausea and he was complaining of abdominal pain. And the only thing that will work on that are opiates, narcotics. And so you can imagine somebody like that has been labeled as a drug addict for emergency rooms in a major metropolitan area. He went for all of them. But between episodes, and I've seen him multiple times between episodes, he's a really sweet guy. He's completely normal. He has no symptoms whatsoever. But during episodes, he has extraordinary pain. I found mutations in the RYR2 calcium channel. I put him on propranolol, and his problems went away. I had previously put him on a mitochondrial cocktail, and he got a lot better. Then he got worse again, and then I did the DNA and then put him on propranolol. I know of 6 different times that he came off of propranolol, because he changed states and he didn't have the authorization anymore, or he left it at home, or he decided he was better and didn't need it anymore. Some combination of those. He just forgot. But 6 different times he came off of propranolol and he relapsed and ended out in emergency rooms again in episodes. So these episodes would last 3 days each. And so he couldn't have a job because every few weeks he would have an episode for 3 days. He would lose his job. And every single time we put him back on propranolol and the mitochondrial cocktail, and he would get better. So that's one situation. Get better, be completely normal until he stopped again. It was a compliance issue. Another one I know is a girl, and she was 10 when I first saw her. She had episodes of very severe dizziness. And the dizziness was so bad that she really couldn't go to school or anything. It got to the point that she was not able to stand. So she was completely disabled by this dizziness. Found an RYR2 mutation in her, put her on propranolol, and then her issue went away completely as well. So that it's it's pain, nausea, vomiting versus dizziness, but it's really just a different symptom that's doing that. A young lady had weird episodes that everybody thought were seizures, and then they did EEGs and they thought they were pseudoseizures. In other words, she's making this up. So of course she loses her job, she loses her insurance, and no one will treat her. She's having these episodes, she's at home and she's miserable and nobody believes her. We found a calcium A1A mutation in her, put her on acetazolamide, and her episodes completely stopped. And she was able to go back to work and live a normal life. So, I mean, there's many episodes, I can go on, but it's finding the exact mutation can make a huge difference in how to treat the patient and how they do, and just in the way they view themselves and how they can live life. But it's complicated.
For the first two examples that you gave, they were both RYR2 genes, but why did that one present as dizziness? Do you have a sense of why one was kind of focused in the gut and one was focused in dizziness?
Because the RYR2 variants are better thought of as polymorphisms. They are variants that are not extremely rare in the population. In fact, the one in the girl is seen in 1% of the population. You say, well, that can't be the cause of disease. And it's not. It's a contributing factor. He has other variants and other genes, some of which I found, some of which I haven't been able to find yet, that do that. So it's a combination of all of her genetics. We are more than just one gene, one variant, and her environment and just who she is and the way that she developed. Identical twins can have personalities which are somewhat different and different diseases just because they developed a little differently. So it's much more complex. I mean, they both had dizziness and they both have nausea and they both had pain. But in one of them, the dizziness was 90% of the problem, and in the other one, the pain was 80% of the problem. So that's how they presented, and that's how they're called.
What does the RYR2 gene do, and how can it be related to functional disease?
Great question. The RYR2 is a calcium channel across the endoplasmic reticulum. The endoplasmic reticulum, if you remember from biology, is this network of like webs that go for the entire cell. It goes everywhere. So calcium is pumped into the endoplasmic reticulum by pumps. And then when the cell needs to do its thing, which in a nerve would be to fire, calcium is released from the endoplasmic reticulum into the cytoplasm, and that tells the cell, okay, it's time to do it. So in a muscle cell, in particular in a cardiac muscle cell, in a heart muscle cell, if you have too much if the RYR2 opens up, you have a bunch of calcium, and the cell contracts hard and fast. So RYR2 is operated in some part by stress. Norepinephrine, the major stress hormone, will cause RYR2 to open up and let calcium out. So the idea is that you see the tiger and that you go into a fight or flight response because your RYR2 calcium channels just open full force. The calcium rushes into the cell, and now your heart rate goes up way high, and your stroke volume, how much you're actually pumping, goes up at the same time. That's the way it's supposed to work. In some patients that have RYR2 mutations, the stress will cause the RYR2 to open up even further than it should, and that the heart cell just keeps pumping so fast that you suddenly die. You get into an arrhythmia. And there are people that have sudden death during exercise or other sorts of excitation with RYR2. But though my patients have never had that issue, what my patients have is the RYR2 opens up probably in the sympathetic nervous system, the fight or flight response, and it causes a fight or flight response. So these patients, they say, I don't feel like this is a problem. This is just a tiny annoyance, but my body feels like it's a problem. I feel very anxious. I feel like there's butterflies. I feel like I'm jumping around. I'm sweaty. I have palpitations. I can feel my heart racing. But it's really just a minor annoyance. And people think it's psychiatric, that they can't handle the tiniest bit of stress. But what it really is that a small amount of norepinephrine that should cause a little bit of calcium release is causing massive calcium release in a total fight or flight response. Propranolol works to block the release of calcium by RYR2. So that's one of the things. But since the mitochondria is on all the time, the mitochondria sort of burn out. And so you get the problem, burnt out mitochondria is what you see in aging. So what do you see in aging? You see functional symptomatology, fatigue, pain, GI issues. And so we need to kind of rehabilitate those mitochondria with a mitochondrial cocktail. So that works as well.
What kind of problems are there in genes beyond channelopathies?
Well, sometimes the problem with energy metabolism is a supply problem, not a demand problem. Not that you're using too much ATP, but you just can't make enough. So we see a lot of genes in the mitochondria. In particular, in the mitochondrial DNA that comes only from the mother, is that we find variants in the mitochondrial DNA which affect energy metabolism. Or they could be in the nuclear genes from either parent. Sometimes they're in the genes that are necessary for peripheral nerves to work. The peripheral nerves are sending the signals of dizziness or nausea or pain from the rest of the body. If those nerves are not working very well, then you can get false signals. So many of the genes are involved in that. In terms of treatment, the energy metabolism is treated with the mitochondrial cocktails we went over, and the second group is treated as neuropathic disorders, gabapentin, amitriptyline, et cetera.
What kind of testing do you need to do to find these genes that are really underlying this functional disease?
Well, the first thing to know is that there are multiple, and in a given individual, it's rare that I find just one variant and one gene, but there are many. So you really have to sequence all of the genes. So that would be whole exome sequencing, or WES. In whole exome sequencing, then I can look at every variant in every gene and then try to put together the picture of which pathways are involved. A lot of times there will be mutations or variants in 2 or 3 of those pathways, the channels, the energy metabolism machinery, and the peripheral nerve genes. And there's other ones I haven't gone on as well. So that's really the important one. In many cases, it's okay to do a singleton, meaning that you're just looking at that patient, not sequencing other relatives. If the disease is extremely severe, in particular, if you have neurodevelopmental issues like autism or ADHD, if you have birth defects of any kinds or dysmorphic facies, the child looks sort of syndromic, then these are often what are called de novo mutations, or new mutations that were not present in the parents and came up in the baby, in the sperm or the egg, or in the early zygote of the embryo. And for those, you need to do a trio. The trio is to sequence mother, father, and the child, regardless of the child's age, and then use a computer to piece it together to find what variants in the child are not present in either parent. And in that situation, we really need to do whole genome sequencing, because I need the genomic architecture to figure out what's going on, to be able to know which, in particular, some large mutations that cover large parts of genes or more than one gene can be there. So if it's functional only, somebody has headaches, fatigue, fibromyalgia, irritable bowel, I usually can do whole exome in the patient only. But if you start getting to severe disease, if you start getting neurodevelopmental problems like autism and birth defects, then we need to do whole genome and we need to do trio.
And to go back to some of our treatments, we've listed some of the pharmaceuticals, we've talked about dietary things, we've talked about fight or flight. Just to summarize that, we can say, which I think is pretty amazing, the way that our minds work and the way if we use meditation or breath work or some of the other things to calm down our sympathetic nervous system, we can actually impact the ways in which our genes express. So that's kind of remarkable. The last thing that I think should touch on today is this piece about the micronutrition and the detoxification and how do we support our mitochondria, because you actually make some pretty targeted products for this.
Yes, I do. I'm now putting on my Neuro Needs hat. I'm one of the founders, and I'm also the scientific and medical director of Neuro Needs, or officer. And Neuro Needs is a company, it's a startup company. There's only 3 of us to make some of these products. I've been using micronutrition or food supplements for 30 years now in patients with mitochondrial dysfunction to treat everything from autism, ADHD, depression, anxiety, pain, fatigue, dizziness, nausea, cyclic vomiting, you name it. And many times it makes a huge difference. But it was very difficult for the families to do this because a lot of the stuff over the counter is completely useless, or it's not bioavailable, or it's in the wrong form. They would have to buy the right products, multiple products from multiple different companies, and then put it all together into the child, which is hard for anybody to take. Somebody that has a life, or a kid who doesn't want to take 20 different capsules, it's difficult to do that. So you have all the fillers of that as well. So I put some products together to try to make this easier. One of the products that I have that's been out for several months now is called Energy Needs. Energy Needs is in capsule form, and it has 40 active ingredients, all natural, that you can find in any food. So it includes vitamins, minerals, antioxidants, and some of the issues that are used to try to detoxify, like glycine and carnitine. Mm-hmm. So 40 different ones. And some of them are very high dosing. That's appropriate when it's safe. And some of them are at lower dosing because I don't want anybody to be harmed by this. So dosing is what I think that pretty much anybody can tolerate without any problem. Mm-hmm. And so this is the micronutrition that your mitochondria needs, as well as detoxification. But it also doubles as a really good micronutrition. So you say that it's like a multivitamin and mineral supplement, but it's got pretty much everything in it. So this is for, adults or older children or anyone who can take the capsules that have any condition that might be caused by mitochondrial dysfunction, including any of the functional disorders that we discussed. There's a similar product that's been out for a few years now called Spectrum Needs, which is pretty much exactly the same thing, but it's in a powder form. It's more aimed towards the autism market and more for kids because it's in a powder form. There's 2 flavors, lemon and berry. And it does include some higher B vitamin levels because that's often useful in autism. Coenzyme Q10 is probably the most important of all of the different compounds. It's in both of those, but the problem with CoQ is that it's an oil. The bioavailable form of CoQ is ubiquinol with an OL at the end. And ubiquinol doesn't really mix with powders very well. So I have ubiquinone in both of those products, but the levels don't go high enough for what I want in my patients. So I recommend additional ubiquinol in my patients. And I make a Q Needs, which has a ubiquinol that is in limonene oil, or the oil of lemon peel, instead of the soy oil. That the soy is the one that almost all of the brands use that you can buy on the internet. And the limonene oil, the preparation allows for very high bioavailability.
Can people take these without testing?
Oh, yes. I usually recommend them when I first see a patient to take while we're waiting for the DNA to come back, because these products are designed for anybody can take it regardless of their age or their medical conditions or what other supplements are on or medications or their genetics. So, it's available on the internet. You don't need a prescription. So I wanted to make these safe for everybody. Now, having said that, if you have significant medical problems, and particularly if you're on a lot of medications, it's probably a good idea to discuss any supplement, not just mine, but any supplement you may want to take with your physician first. Absolutely. Just to make sure it doesn't interact with your medications or your medical conditions. But I do recommend them, and a lot of people do take them just because they have migraine or autism or fatigue or something, or just because they've gotten to a certain age that functional symptoms are universal.
Yes.
I take it.
Yes. And how do you pick whether you should take Spectrum or Energy Needs, or is it okay to take both?
Well, they're very similar. It's really if you want a powder or if you want a capsule. Most adults would prefer capsules. Some people have trouble swallowing. And I do recommend taking Q Needs with either one of those to give additional CoQ.
Well, we have really covered the gamut today. We have really gone, when we started out this podcast talking about how we really wanted to go soup to nuts on genetics, and the way that you approach genetics is, I think, still somewhat unique, but becoming more and more, hopefully more and more common. And that, again, you're not really just focused on structural abnormalities, but really the functional aspect of how genes are expressed, the functional aspect of how genetics works together throughout the body, and how it can underlie functional disease. Is there any last things you'd like to say before we close today?
No, I think that we covered everything, and I wonder if I blew everyone's mind or not. For those that are interested in DNA testing, you can contact Neurabilities, Neurabilities, Neurabilities.com, and ask for the neurogenomics program.
Thank you. Well, thank you for listening today with Dr. Richard Boles. We've got lots of ways to continue the conversation. We do want to hear if he has blown your mind. Is that you've definitely blown my mind. We can continue this conversation with through Facebook, Instagram, and Twitter. You can get more information about Dr. Boles and his products at Neuro Needs, NeuroNeeds.com. And more info about and from us at our website, centerforhealingneurology.com. Please be sure to share this show with your friends, and we welcome your rating and review wherever you get your podcasts. And feel free to send topic requests to podcast@centerforhealingneurology.com. We love that you've joined us today to discuss how to make our whole world medicine. We rise or fall together, and we're committed to doing what we can to make as many of us as healthy as possible. And this takes all of us, including you. Thank you for listening, and see you next time.
Partyfish Media.
Partyfish Media acknowledges that it operates and records on indigenous Duwamish and Puget Sound Coast Salish land that is still home to the Duwamish tribe. This land is stolen in violation of the Point Elliott Treaty of 1855. We are committed to uplifting the name of these lands and community members from these nations who reside alongside us. For more information on this land, its people, or ways you can help, visit duwamishtribe.org or realrentduwamish.org.